This is an OCI liver biology video continuing my work from Unit 5.2. Now we're looking at the kidney. We did the liver in the previous lesson, but let's get on to this. So there are two kidneys, and both these kidneys will have a renal artery, a renal vein, and this structure. So this is one kidney drawn out here. We've got a renal artery, which is the red one, and a renal vein, which is our blue one. So the renal artery obviously brings in oxygenated blood, and the renal vein takes in this not deoxygenated blood, but filtered blood from our kidney. So we've got these three layers. We've got the cortex, the medulla, and the pelvis. So the cortex is the outer layer, the medulla is the middle, and the pelvis is the bit containing our renal artery and veins, which basically our fluid flows into and is extracted. We've also got this here, which is our ureter. So basically the urine fills up in our pelvis and ends up going to our bladder and to be expelled from the body. So within our kidney, by the way, we've got this nephron structure. So I've drawn it really small here. Later you'll see it. You should know what I'm talking about from GCSE knowledge anyway. But this is how the nephron would look. So we've got the Bowman's capsule here, proximal convoluted tubule in the cortex. Our loop of Henle reaches into the medulla, and then the rest of the nephron is in the cortex. However, some of the collecting duct will reach into our medulla. So there's 1 million nephrons per kidney, but let's look at one nephron starting off with the glomerulus and Bowman's capsule. So the glomerulus is basically a network of blood capillaries. Going into it, we've got this afferent arteriole, which is much wider than our efferent arteriole. At the end of the video, I'll speak about our GFR rate, glomerular filtration rate, and how these two things can affect that. But just remember, afferent is wider than efferent. We've got some other things in between our podocytes and glomerulus, which I'm going to draw in much more detail after, but let's just get the rest of the Bowman's capsule drawn on. So here are in blue our podocytes, which I'm about to expand on here, and we've also got this epithelium of the Bowman's capsule around the outside, and our proximal convoluted tubule and epithelial cells lining that. Okay, so let's look at podocytes, basement membrane, and our capillary endothelium. Okay, so within our capillary endothelium we've got lots of pores, and we call these pores fenestrations. They allow blood plasma and dissolved substances to pass through. So fenestrations in our capillary endothelium allow plasma and other things to pass through. Then we've got this yellow basement membrane. It's very thin. It's a fine mesh of collagen fibres and glycoproteins. The mesh acts as a filter to prevent the passage of molecules that are larger than 69,000 of their molecular mass. So a molecular mass of 69,000 moles. So such as protein and blood cells, they cannot pass through our plasma membrane. Our basement membrane. So the basement membrane blocks out these large substances. Fenestrations in the capillary endothelium allow substances to move through into the Bowman's capsule. Then we've got these podocyte cells, and podocytes have finger-like projections called major processes, which contain minor processes, holding the cells away from the endothelium of the capillary, ensuring there are gaps to allow blood to pass through into the lumen of the Bowman's capsule. Okay, so these major processes hold the cells away from the endothelium of the capillary, ensuring gaps. Okay, so just a little summary here. We've got endothelium with fenestrations allowing blood plasma plasma to pass through and other solutes to pass through with it. Then we've got our basement membrane, which blocks substances which are larger than 69,000 moles, so proteins, blood plasma proteins cannot pass through, and that's a good thing. They keep that oncotic potential within the blood, in unit 3.2 or 3. And finally we've got this epithelial cells, which is mainly composed of podocytes, which just hold the blood capillaries in place with many finger-like projections, holding cells away from the endothelium of the capillary. So this afferent efferent thing helps maintain a high pressure in the glomerulus. Like I said, I'll speak to you about GFR rate at the end of the video. So what passes through in our glomerular filtrate? We've got these substances: water, salts, glucose, urea and mineral ions. So all these substances pass out and we end up with a very low water potential in our capillaries through the efferent arteriole. So one thing I need to speak about here is this process, and I've written it in large letters here, is called ultrafiltration, which is the filtering of blood at the molecular level, caused by all these different features, caused by the high pressure in our glomerulus, just the blood being in contact with our basement membrane, podocytes, okay, the difference in potentials between the glomerulus and the Bowman's capsule maintains high concentration gradients, and therefore substances move out from the blood into the nephron. Blood cells and proteins are left in the capillary, giving blood a very negative water potential, ensuring some fluid remains in the blood. What we've got here is, a picture, light micrograph of a kidney. So we've got here a distal convoluted tubule, which I'm about to come on to. We've got our space of the Bowman's capsule, so this space here, and the glomerulus inside here. So erythrocytes are running through here. This is our glomerulus. Bowman's capsule space, which is surrounding here. Proximal convoluted tubules are little tubules like this. So that is a cross section. You need to sort of recognise different features within histology. So let's move on to our nephron, and I'm sure you're going to recognise it from GCSE. I'm going to draw it out nice and quickly now. This is as sped up as I can do. So you've got your Bowman's capsule. I copied and pasted it, and some bits were missing, so I had to draw them back on. And then we've got this, our proximal convoluted tubule, loop of Henle, distal convoluted tubule, and then our collecting duct, which has lots of nephrons joined together in our collecting duct. We don't just have one. Remember, we've got millions of nephrons in our kidneys, in each kidney, and they join at different points. Okay, and that is no issue. One collecting duct can join many nephrons together. And we've got these two layers, so our cortex and medulla. If you remember at the start of the video that I spoke about these different layers of the kidney, the cortex being the outermost layer, the medulla being the middle. And we're ignoring the pelvis for this. The pelvis is not really involved. This will end up being transported all the way to the pelvis in the collecting duct. So let's take each bit step by step. We've looked at the Bowman's capsule. Now let's look at our proximal convoluted tubule. So within the proximal convoluted tubule, selective reabsorption happens. So selective reabsorption basically is meaning we're selectively reabsorbing. I don't know how to define it without really using the words. Just selecting sort of compounds like glucose, water, sodium ions, things we want in the blood that we filtered out of the nephron, we want back in the blood. We don't want to lose all of these things. So glucose, any proteins we lost, can also get back into our blood, and they move via endocytosis. So they come in here via endocytosis and into the blood. Okay, so the centre bit is our tubule. So we've got lots of solutes and liquid flowing through our tubule here. So on the outside we've got these cells. The cell surface membrane is in contact with the tubule fluid, and it's hydrally folded to form microvilli, which increases surface area for reabsorption. At the opposite cell membrane, we've got sodium potassium pumps, which you should recognise from unit 5.3, which is nervous impulses. So sodium potassium pumps, they pump potassium in from the blood and pump sodium out of the cell into the blood. So potassium moves in and sodium moves out. So we get a low concentration of sodium in our cell as we're pumping it out. Therefore, sodium, which is contained in the tubules, diffuses in to our cell. So obviously sodium wants to move back into the cell, but the only way sodium can get back into the cell is through co-transporter proteins, which requires sodium to carry with it either glucose or an amino acid. In this example I use glucose, but just remember amino acids can be carried through these co-transporter proteins with sodium as well. So this whole selective reabsorption process, we're basically looking at sodium, amino acids, glucose and water, which I'll explain what water follows through in a second. So sodium and glucose come in here. We end up getting a high concentration of glucose within our cell, and I'll just draw on the potassium being pumped into the cell, which I forgot to mention because I did tell you these are sodium potassium pumps. Just ignore that potassium for now though. Pretty insignificant here. So this high concentration of glucose in the cell causes water to be drawn in, because this high concentration of glucose causes a low water potential. So we've got a higher water potential outside the cell than inside the cell, so water rushes in and follows our glucose, and that we see that happen countless of times. Okay, so let's split this down really nice and quick. So potassium is pumped into the cell, and for the potassium pumped in, sodium is pumped out. Okay, this causes a low concentration of sodium in the cell. Therefore, sodium moves from the high concentration in the tubule to the low concentration within the cell, but the only way it can do that is through co-transport proteins, which requires it to carry glucose or amino acids. So for this example we use glucose. It diffuses into the cell carrying with it glucose. The glucose builds up. We end up getting a high concentration of glucose but a low water potential because the concentration of glucose is so high. Water moves from a high water potential to a low water potential, therefore water follows this sucrose and sodium into the cell. Then the cell ends up having a higher water potential and a higher sucrose concentration, as the blood flowing through our kidney has a very low water potential, remember, because we just did ultrafiltration at the Bowman's capsule. Therefore these capillaries have low water potential and low concentration of all these solutes. Therefore everything diffuses into the blood through the fenestrations of the endothelium. So that is selective reabsorption. Now let's look at the loop of Henle and its role as a countercurrent multiplier mechanism to so the loop of Henle has a really important role to basically set up the right gradients, the right water potential, both of the tubule and the medulla. So when it gets to the collecting duct, when the fluid gets to the collecting duct, more water is reabsorbed by the blood because we've set up this countercurrent multiplier mechanism. So let's break it down slowly we've got a descending limb and an ascending limb, and I'm just going to hollow these limbs out just to make it easy for me to write on. So the ascending limb is impermeable to water, and in the ascending limb, sodium and chloride ions are actively pumped out through pumps. So this is an active process, it requires ATP. They're actively pumped out of the tubule into the medulla. So we end up getting a high concentration of sodium and chloride in our medulla. So as water moves down, so as the tubular fluid moves down the descending limb, it starts with a high water potential here. High water potential in the tubule. Then it meets this low water potential in the medulla. Okay, so obviously water moves from the high to the low water potential, so water moves out of the tubule via osmosis. So what happens is these mineral ions that we've pumped out end up diffusing into the descending limb, as we've got a high water potential here, so a low concentration of these mineral ions here, so they naturally diffuse back in. So as the water moves, as the tubular fluid moves down, we end up getting a lower water potential here than we do here. However, it's still higher than our medulla, as the medulla is having sodium ions pumped into it. So the water potential at the bottom of the descending limb is still higher than the medulla for it not to want to move out by osmosis, but it is much lower than what it started at the top of the descending limb. So I'll draw that on here. Top of the ascending limb, high water potential. Bottom we have a low water potential. It then moves around to the ascending limb, which is impermeable to water. Mineral ions move out of the ascending limb via diffusion at the bottom because they've got a high concentration gradient now, as we've got this low water potential. They diffuse out. There's further active pumping of mineral ions out, but at the end of it, because of this diffusion of sodium and chloride ions out, active pumping of all the sodium chloride basically, nearly all removed from our tubular fluid, we end up getting a high water potential here at the end. So what we've managed to do is give the medulla a really low water potential which surrounds our collecting duct, but we've given the tubular fluid a high water potential, not as high as it was, but a high water potential. We've managed to maintain that. So in our distal convoluted tubule, we see active transport take place, and just basically regulating the mineral ion contents. And I'll write that on later. So it's not something that you're likely to get tested on distal convoluted tubule. The book pretty much bypasses it. It's not really in our specification, so we can look at that. We call this a hairpin countercurrent multiplying mechanism because of what I've told you about. We end up getting this high water potential in our tubule, a really low water potential in our medulla. It's creating this concentration gradient for water to be reabsorbed. So it's a water potential gradient for water to be reabsorbed in the medulla. So quite overkill with that. Let's just look at our collecting duct here. So water gets into the top of collecting duct. It's got a high water potential. It moves down the water potential gradient. Water constantly moves out into the medulla with a very low water potential, and any water that's not reabsorbed, because we've got this surrounding capillary network here, we're surrounded by capillaries. The water is reabsorbed into those capillaries, and then any water that's not reabsorbed then goes into the urine. the permeability of the collecting duct can be altered, okay, and I'm not going to go into that right now, but I do look at it later. And looking at how long we've been on this video, it will be in a third video. But one last thing, just because we've covered the whole nephron, this is the last thing really on the nephron that we want to look at, and it's the changes in pressure in each of our chambers. Not chamber, changes in pressure in each of our for each of our substances. So first of all, we look at glucose, and as we see, all of them, by the way, start at concentration of 1. Glucose enters the proximal convoluted tubule, and all of it is reabsorbed. So by the end of the proximal convoluted tubule, we've got about 0.02, which is a negligible amount of glucose. Okay, it could be even less, but nearly all of our glucose is reabsorbed there. Okay, so we can ignore that. We call that selective reabsorption in the proximal convoluted tubule. Next, we look at our mineral ions, both sodium and potassium. So nothing happens to them during the proximal convoluted tubule. In the loop of Henle, remember, first it goes down the descending limb, they diffuse into the descending limb. In the ascending limb, they are actively pumped out, so we see it shoot down. By the end of the loop of Henle, we've got this much lower concentration. But in the distal convoluted tubule, we see active transport, and we see the concentrations regulated, so that potassium shoots up and then sodium shoots up as well. Okay, so let's finally look at urea, and urea is our purple line, and the main reason for the increase in urea concentration is because water is being reabsorbed. So as water is reabsorbed in our proximal convoluted tubule, water moves out of the descending loop of Henle. The concentration therefore will rise because water has been selectively reabsorbed. And we see active movement of urea into tubules, okay? Right, so that is all on changes in concentration. That is the whole of the nephron. There is more to come, and I just did something. I accidentally undid a step, and the half of the title has now disappeared, but that does say osmoregulation, and that is what we're going to be looking at next. Obviously this video has been a long one. This will be the final video, osmoregulation. If you really want to watch it next. Thanks for watching this.